My pleasure today to welcome Dennis Whigham here from the Smithsonian Environmental Research Center. I first knew of Dennis when I was a graduate student. I was looking at tidal freshwater wetland vegetation, read some of his papers. He's been at the Smithsonian, I think, since 1977, over in Edgewater. First as a botanist, then as a senior botanist, and retired last year and now is emeritus scientist emeritus. But he's doesn't just work in wetland plants. He works in he's an expert in orchids. He started the North American Orchid Conservation Center. Google it. And so they've done a lot of amazing research with orchids and how they reproduce, really insightful stuff, plant ecology, wetlands. And he was at Rider College actually for a few years before that. That's where he started doing tidal freshwater wetlands. I believe you have your website at the CERC says more than 235 papers, journal articles, maybe pushing 250 by now. We worked with Sylvia Jacobson. He was on her committee, and we've done this work, and he's working with Jared and me. And so really glad to have him here. I will say he's been a mentor to me over the years as somebody who's done an incredible amount of things, somebody I've looked up to and become a friend. And so we go have coffee about once a month, sporadically, and talk about stuff and things that are going on. He's still really active since he retired, and this is a transition, but really an amazing person. And I think you might see a glimpse of his potential for delving into all kinds of areas with this talk today. I haven't heard a lot of this. I've heard bits and pieces of it, but something different. So, Dennis, it's really great to have you here. Thank you for coming to talk with us today.
Okay, thank you, Andy. When I guess it was Sylvia asked me if I'd come over and talk, I could have talked about Phragmites. You've probably heard about that. You know, here in Phragmites. We could talk about orchids. That's kind of a little bit away from here. And what I'm going to talk about today is probably one of the most satisfying projects I was involved in over almost 50 years. So I'm going to give you some background as to how we ended up in Alaska and doing the kinds of things I'm going to tell you about. The first slide clearly demonstrates that this isn't just me. I sort of had the idea to get this going, but it's involved a bunch of other people, particularly Ryan King at Baylor University. He's now retired. Cooey Walker, I'm going to talk more about her. She was the initial link. These are people at the National Estuarine Research Reserve. We just heard Sylvia talking about the National Estuarine Research Reserve in Maryland. This is the one that's up, the only one in Alaska. And then the bottom line there is Mark Rains, a guy that I've worked with for many, many, many years. He's sort of an eco-hydrologist at the University of South Florida. He and I worked on mangroves and wetlands and all kinds of things. And he's still very actively involved in this project in Alaska. I'm sort of not quite faded out, but close. So to set the tone, I don't know, how many have been to Alaska? Anybody? Got a few here? Okay, I'm going to set the tone with the place and kind of why we're there, and then I'll go back to the beginning. Can you lower the lights a little bit?
Not everybody in Alaska is a fisherman, but all of us live on salmon land. The local farmers, they have little salmon streams that
This is
go
Cooey
through
Walker
their property.
talking now.
If you look at a map of the Kenai Peninsula, it just looks like a cardiovascular system. Salmon are like the heartbeat.
The Salmon and People Project is a combination of understanding basic science and ecology and communicating that information to decision makers, to people, to the public. And so the whole goal of this is to understand how the ecosystems work here so that people have the option of making good decisions when they decide to do something on the landscape that won't degrade the resource. Everybody here loves salmon and they want sustainable salmon. And we hope to give them the information that will enable them to do that.
Trying to find a hole that we got last period.
There was something that came alive in me when I went out fishing that I hadn't really felt before. It was being connected to something that was bigger than myself and bigger than my 23-year-old girl problems, right? And it was the first time that I really felt that I could be part of a landscape and a people that I was proud of.
My father brought up 3 boys hunting and fishing and not throwing stuff away and not throwing garbage down, and so we slowly kind of developed into humans that took care of those resources. When you come home from work or you go through a divorce or the normal stresses in life, when I go down to the river takes all those stresses from me. I just don't think about other things, watch over your shoulders for bears, and enjoy that nature, because you live in a place where it's available to you, where you can walk to the river from here and walk to the beach from here. Everybody in life's probably looking for their idea of this. Here, I feel it's our place to fight tooth and nail, just to keep it healthy.
Everything about salmon is exciting to me. Right now I'm working with salmon vertebrae, and my process is gathering, processing it, and figuring out what I want to say with that material. For me, art has become so much more than object making. It's incredibly important that we feel connected, because we are connected. If we lose that sense, we lose our connection to our habitat.
The last few years have been really slow in cooking lake. And to a certain degree, that's normal for salmon. They have big boom and bust life cycles, and as fishermen, we are really used to just riding those out. But lately, it's been really, really bad fishing. Most of our fishing days this year have been really hot, and there's a theory that fish are swimming deeper, swimming in different patterns, and so we're not intercepting them. Temperature becomes a big problem too once they get up the river. Are the rivers cold enough to sustain those juvenile salmon? So is this part of our fish run's natural cycle, or is this related to a larger climate shift that we need to be worried about? We have more questions than answers about what's happening to our salmon here. It's been nerve-wracking to watch.
My name is Kuli Walker. I'm the manager of the Ketchikan Bay National Estuary Research Reserve, and Smithsonian is funding us and helping us connect with our communities, connect the science we're doing to the communities where those decisions and that science can be useful. What we've been doing over the past years is understanding how the baby salmon, which are living in the far reaches of the watersheds, how the landscape is supporting those streams, which is supporting those juvenile salmon, which most of us have never seen before. So today what we're going to do is just sort them, ID them, and collect stomach samples so we can see what they've been eating. What they've been eating, what we've learned, is directly related to the nutrients coming off the landscape. You know, that's the message we carry out. It's like, how do we conserve the landscape so that it's still supporting the food that the salmon eat and the salmon themselves. Let's find the plumpest fish we can. We all have to understand that what we do in the landscape around these streams is having an effect on those streams, an effect on those baby salmon, people all of a sudden go, oh, I don't want to hurt salmon, I want salmon to be there for, generations to come. But we have to make the right decisions as a group, or we won't have salmon.
So my goal in this is to see that salmon remain a sustainable resource here in Alaska. And one of the key things we've learned from this project is the linkage between the land, the wetlands, and the salmon. And if you degrade those resources, it's going to affect everything. When the water flows off of the land, it passes through the wetland before it gets to the stream, so that any kind of interaction between the water up there and the stream as it passes through the wetland will influence the ecology of the stream. When these plants die, they're going to fall over and hang over the bank of the creek, and then that becomes a source of organic matter for the creek itself. So we're going to measure that also. We have made a lot of mistakes around the world in how we've managed and changed ecosystems, and there have been huge consequences to that. We spend billions of dollars trying to fix things that we made a mess of. It would be nice to be able to take information, provide it to people so that they can still earn money, do things, but do it in a way that is not altering the ecosystem and damaging it. And this remains one of the few places where we can do that.
Those things are changing, and maybe permanently. Anything we can do to understand how those changes are affecting fish and how those changes are affecting the people that depend on fish will be great. More and more I've seen fishermen and scientists really working closely together and considering themselves part of the same team. That makes me think that we can weather bigger shifts as they come.
Salmon is the heart of the ecosystem here, and it's the heart of people too. So I'm just determined to do everything I can. Our partnership with the Smithsonian and the researchers are make sure that we can do whatever we can to keep things connected.
The more I realize how much resource I use, the more I realize that I should give something back. And if you don't do something up here as an individual, then eventually they'll go the way that the resources have gone other places. Then you just have a pretty place left, but nothing for fools like me to do.
Okay, so that tells a story. We're done. You can leave now. So how did we get there, and what's some of the science behind what you heard about? So this is the outline. I'm going to give some background, talk about some of the landscape studies that we did, and then a few experiments. And so this wasn't initially a hypothesis that we set to answer right at the beginning, but as we learned, then we sort of developed ideas and did some things. And I'm going to end with this, with, okay, we've done some science, we've published a bunch of papers. Is anybody doing anything about it? And those of you who've been in this business a long time know that's the hard part. Conveying our science to people that have to make decisions. So let's talk about the background. Turns out this study actually started here in the Chesapeake Bay. Many years ago, we were involved with a bunch of other universities and folks in a very large, multi-million-dollar EPA-funded project called the Atlantic Slope Consortium. It's directed by Rob Brooks at Penn State, who's passed away since then, and Denise Warder. On the left, you see a map of the bay, and what you're looking at are watersheds that drain into the Chesapeake, and you'll notice they're color-coded. And the idea behind this project was to take different kinds of watersheds, some developed, some agricultural, some mostly forested, and ask, when humans start changing things on the land, what happens in the water? And so we had people measuring water quality and PCBs and fish and Phragmites and all kinds of things. And the most striking result of that research project, which was a 5-year project, is really in this slide. The bottom of this shows how much of a watershed is developed from, nothing to 100%. On the left is the probability of something changing, and you're looking at some of the variables that we measured. For example, the abundance of Phragmites is one of those things. And you'll notice for all of these indices that when the land begins to change, the metric for that variable begins to change, and by the time you've reached about 20% of the landscape being altered, you have a changed ecosystem. It's different. Everything has changed. And we spent billions of dollars trying to repair the Chesapeake Bay. We're never going to get it back to what it used to be because of this. I mean, there are just so many people here doing so many things, it's not going to become original. We're just trying to hold it to where it was. And the other thing we found is that when these metrics were changing, the closer you were to the shoreline, the faster and the more the metric changed. So when we do things close to the shore, that's even as bad, if not worse, than just doing it somewhere else up on the watershed. So with that in mind, I started to think about, well, can we go to some place where we haven't screwed it up, find out how it works, and put that information together in a way that can help people make decisions that will result in preserving what they have, at least not messing it up the way we have back here. And I had a connection in Homer, Alaska, which is you see down here. This is Homer. And my daughter lives there. She's lived there for 25 years, so I had a reason to go there. But even before then, I had contacts up there. And one of the contacts was Cooey Walker, who you saw in the video. Here's Cooey. And and she was a student of Rob Brooks's, one of his first students when he wasn't even on the main campus at Penn State. So I knew Cooey. Little side note, Cooey's father died 2 years ago, 94, 95 years old. He was a distinguished music professor at this university. An incredible man. Spoke 5, 6 languages, worked all over the world. He wrote music. He is just an incredible guy. She's incredible too. And so Cooey was there, and so I contacted her and I said, Cooey, I sent her some papers and I said, we've been doing this stuff back here, and how about, is there anything we can do up there that might work? And she said, yes, I'm starting a landscape effort within the National Estuarine Reserve Program here, and we've noticed that there are a lot of tiny little fish in tiny little streams, but we don't know anything about them. And that was the spark. And so she and I worked that idea a little bit, and I said, well, I'm just a dumb old plant ecologist who studies wetlands. We need to get somebody else involved in this. And so I got Ryan King to go up on a trip to Homer. Ryan is, as I mentioned, a professor at Baylor, now retired, but the dude can do anything. He teaches statistical courses, he's an expert fly fisherman, and he just does it all. He's a really incredible guy. So coming to Alaska to maybe see fish, the dude was on it right away. So he and I went up there and met Cooey, and we looked at these streams, and they put a little trap in there, and the stream was not any wider than this, and there were all these little fish in there. So we put our heads together. We ended up getting some money from EPA to start a project. So here's a little bit of what the Kenai lowlands look like. Over on the right, this is a sort of a fluvial, glacial moraine kind of environment. Mountains off to the right, the Kenai Mountains are over here. And there are a lot of different kinds of wetland systems here, and you can see them on the right, and they're spatially distributed across the landscape. And over on the left are the streams that we ended up working in, the rivers. None of them are huge rivers, but big enough to have adult salmon come back into them. There are 5 of them, and you'll notice 2 things on that diagram on the left. The blue lines are places where salmon are known to have been, adults coming back from the ocean. And when the state recognizes that water is protected because it has salmon. But you'll notice there are a lot of red lines there. These are headwater streams, first and second order streams that had not been sampled. And as far as the state of Alaska was concerned, they don't have any protection. And if you look at the length of the red lines compared to the blue, you see there are way more red lines on the landscape than there are blue lines. And so the red lines were the focus of what we were going to look at. And the red dots that you see on the left, these are sites that we sampled over a 3-summer period. I was in the best shape of my life then, lost like 25 pounds, carrying 50-pound backpacks across the marsh, across the landscape, to look at these kind of streams. And I'm just showing you kind of what they look like here. Some of them are so overgrown by grasses that you hear the water, but you don't see the water. So this is kind of an example. There's beaver there. Obviously, also there's Steve Baird, one of the guys who worked with us at the reserve. So let's talk about the initial landscape studies. We didn't know how this place worked, so that was the first job, was to go up there and collect a bunch of data. So we went to all those sites that had the red dots. Here's a picture of some of the things we did. We did geomorphology, we measured the vegetation, we measured the morphology of the creek, we did water quality. On the lower left, we did invertebrate sampling, and on the right, electrofishing to get the fish. So we were basically looking at everything we could find to try to put it together. And now I'm going to show you some data. And so when we take all the data that we gathered and threw it in there and did an ordination of it, this is what it looked like, but it was as clear as a bell. That is, we have basically two kinds of systems up there. We have high gradient watersheds that were dominated by riparian wetlands. Those are the ones on the right in the blue. And over on the left are low gradient wetlands that are dominated by basically vast wetlands, drainage ways, lake beds, kettle wetlands. So the water's moving more slowly, it's more flat there. And that's a fairly clear differentiation of, based on a lot of data. So that was a real help to us. We had these two categories of wetlands that we could focus on. And that's meaningful. So for example, if you look at the slope of the land, that's what's the x-axis here in all these. I'll just go through a couple of these. So first of all, the slower, up here in the upper right, if the water's moving slow and it's sitting on the land longer, temperature of that water is going to be higher than it is if it's basically groundwater coming off of a steep slope, okay? If you look at dissolved organic nitrogen, so the peatland, these slow-moving systems on the left, there's not much nitrate there basically, but over on the right, that's groundwater coming off, and I'm going to get into that for other reasons, there's more of it. So you can see from these graphs that there were really differences between the slow-moving systems and the ones that are on slopes, even though, like most things in the world, they're along a gradient, of course. If you look at the invertebrates that were in that stream, it was the same sort of result. Oh, I'm sorry. We basically had two groups. Over on the right, where the blue things are high gradient streams, and they have their own suite of invertebrates that you would find there. Mayflies, stoneflies, blackflies, free-living caddisflies. Over on the left, the more slow-moving systems, they ordinated over there, and these are different things. Snails, segmented worms, caddisflies, things like that. So again, the animal world is separating also into these two kinds of systems. On the plant side, it turns out there's one dominant wetland grass there that's not just a wetland grass, it occurs all over the landscape, and it's a blue joint, Calamagrostis canadensis. It occurs in this world too. And you can see from the importance value there on the top right, that was really the dominant system. To the point where if you looked at the diversity of wetland species that are associated with these streams as it related to the abundance, the importance of Calamagrostis, you see a clear relationship there. As Calamagrostis became more and more abundant, and higher density, things like that, you had fewer wetland species associated with it. And that becomes important, and we did a lot of studies with the Calamagrostis. The upper left is one of the studies where we went to 3 different locations along the same stream. Of course, we replicated the streams, and we were interested in whether there's any longitudinal patterns in the abundance of the grass and the biomass of the grass. And you can see there is a little bit of difference there, but nothing that stands out. And that there was a slight difference on the how much Calamagrostis cover there was on the creek bank over here on the left, and a difference between the high gradient and the low gradient stream. So, functionally it was going a little bit different depending on where you are in the landscape. But probably more important was that in the video you saw that this Calamagrostis at the end of the growing season, it flops over. And it's hanging over the creek bank. And that previous image you see that over here, probably 2 or 3 years worth of Calamagrostis hanging over the creek bank. And here it is. And on the right-hand side you can see that some of it's dry, further up on the bank, some of it's wet. Well, what is this? Well, it's a carbon source to the stream. It's a source of nutrients for the stream and the organisms in the stream. It helps stabilize the bank of the creek, and it provides habitat for organisms, and particularly for the fish. They really hang out under there. We also did work with the fish, as I've mentioned, and it turned out they sort of fell out into buckets also based on the gradient of the stream. In the high gradient systems that were on steep slopes, they were dominated by little tiny dollywart. And dollywart is a fish that goes to the ocean and comes back. It goes back and forth. It's kind of like an Atlantic salmon. At the other end, the slow-moving streams, they were dominated by large coho salmon, what they call par before they go out to the ocean. So the fish were basically segregating themselves based on the kind of stream system they were in. And I should mention that when the adult fish come back from the ocean, they can only go so far, and then they lay their eggs, and the eggs hatch, and then the little ones of the coho and a few others, they go upstream and they live in these creeks for 2 to 3 years before they out-migrate to the ocean. So these tiny streams are really important habitats. And they're safer up there, basically. The streams are smaller, that there aren't so many things that are going to try to eat the fish. Ryan did some predictive modeling from these landscape features, and he was able to do a couple of things. Number one is he was able to demonstrate that this landscape full of first-order streams is really important salmon habitat. If the juveniles don't survive, obviously you don't have big ones coming back from the ocean. So these are really critical habitats. The other thing he was able to do was to combine these landscape features with some other features I'll talk about, and he could model where there were hot spots on the landscape, where you would expect to find the highest diversity of fish, which that was also very useful and helpful as people were learning how to take this information and use it. Now I'm going to switch over and I'm going to talk about alder now. So almus is a species that occurs around here. It's global, and the alder, you probably know, it has bacteria in its roots that fix atmospheric nitrogen and put it into the system so that it's then subsequently available not only to the alder, but to other organisms once roots die and nitrogen gets out of the alder roots. So Becky Shaftel, she was a technician the first summer we were there. She ended up doing a master's with Ryan on the role of alder in the landscape. And so what you're looking at here on the left is a map showing the sites that we sampled as part of this alder project. And we selected sites where we knew there was a lot of alder on the landscape, to no alder, to some smaller amount of alder. And here's an example of that. Here are two of the watersheds that she looked at. The one on the right has less alder than the one on the left. So the question is the amount of alder that's on the watershed important to the characteristics of the water in the stream? And it turns out it's very important. And here we're showing the relationship between the percent alder cover and nitrate in the stream. And up here in the top part of this diagram, this is May when the snow has just melted. And so basically everything's sort of coming off of the landscape, and you can see that's a real linear relationship there. But as you get into the warmer months of the year, into July and September, when the snow has already melted, and the water that is coming into the stream is the water that's coming through the ground. It's groundwater, shallow and deeper groundwater that is downslope of the alder. So the more alder you have on the watershed, the more nitrate you're going to get to the stream. Another part of this was to ask whether there's any connection between the alder on the landscape and what the fish are composed of, what their little bodies have in it. And Matt Descartes came onto the project for a year as a postdoc, and he was an isotope guy. So they went out and they collected a lot of samples from these two streams. And they collected consumer organisms, and he did a nice isotope analysis on them. He collected algae on rocks and things like that, and calamagrostis, and he looked at the isotopic signature of those things. And then he collected fish Here are data for 4 fish, a coho salmon on the left, and 3 different Dolly Vardens here on the right. And so he used mixing isotope models with hydrogen isotope, carbon isotope, nitrogen isotope to ask, can you just differentiate what these fish are eating? And it turns out that if you look at the consumer organism in the stream and compare them to the fish, that most of the signature, the squares up on the top, this is an indication that it is a loctinous material. It's coming from outside the stream. And the circles down here in the bottom, this is a loctinous material, meaning it's coming from in the stream. And the bottom line on this is that these fish are basically living off of the landscape. About 65% of the nitrogen, he estimated, was in the bodies of the fish, was coming from the landscape, not from in the stream. So again, the connection to the land that separates it from the stream itself. So based on these earlier studies, we did some experimental studies. We did a dosing study where we hypothesized that areas that had a lot of wetlands, they were getting dissolved organic carbon into the stream, and areas that had alder on the landscape were getting nitrogen. And so, you could figure out that one of them is limiting somewhere. So we did a nitrogen dosing experiment and a carbon dosing experiment. Unfortunately, the nitrogen experiment, the place we did it, had a massive rain event and just blew the experiment off out of the creek. But the one where we did the carbon dosing worked, and I'm going to show you some results from that to show how important these limiting nutrients are to the ecology of the stream. So our hypothesis was that the headwater streams that have alder, those streams are going to be carbon limited because they're getting nitrogen but not much carbon. The headwater streams that have a lot of wetlands associated with them, peatlands, that kind of thing, and not any alder or very little alder, they're going to be nitrogen limited because they don't have any nitrogen coming down from the watershed. So here are some of the data that sort of back why we did that. So if you're looking at, in this middle diagram at slope again, how steep it is, what you find is if you have increasing slope, you have less dissolved organic carbon coming into the stream. If you have slow-moving systems over here on the left, you have more dissolved organic carbon coming into the stream. So that's sort of data behind that idea. On the right-hand side, you're looking at alder cover again over here, and the more alder you have, the more dissolved organic nitrogen you have. So this is sort of the data that backed up the 2 hypotheses that I mentioned. So here are results from the carbon dosing experiment, and this was a little different than most people who have done these kind of experiments because typically in the past what they've done is they've dosed the hell out of it. They put concentrations in that you would never get, in nature, or up to very high level. What we did, because we had the carbon data, the spiking that we did was at background levels for this system. So here's an example of the experiment, or here's not an example, here is the experiment. We had a dosing station, I'll show you a picture of that in a minute. And then upslope of that, we had a treatment reach in which we weren't adding carbon. So we added carbon at the dosing, at the dosing station, and then we measured it for about 75 meters downstream. We had sampling stations along it, you see that in the bottom diagram. And then we had another reference reach, which was sort of beyond where we thought the impacts of adding the carbon would be. And here's a stream that was dosed over here on the right. And this is an example of, we backpacked this all in from the road up on the top of the hill, and we used acetate. Here's the tank with the acetate in it, and we have a battery-powered pump that went drip, 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 and we were putting in 0.25 milligrams per liter just continuously into this stream. And we had a team of people measuring things for about 2 months, and they were measuring metabolism in the stream, nutrient uptake in the stream, the invertebrates that were in the stream, and we measured what was going on with the fish that were in the stream. So I'll show you some results from that. So over on the left is pre-dose. This is respiration, and with increasing respiration, the numbers become more negative. And what this clearly shows is that within 10 days of the dosing starting, you are having a big increase in stream restoration, stream metabolism. And that continued for pretty much the duration of the study. Down here is a gross primary production in the stream, oxygen production. Before, nothing. And then after that, you saw it started to spike within 10 days, but you noticed that it didn't do anything in the subsequent days. And the reason for that is that the Calamagrostis was growing this tall and shading out the stream. So the light wasn't getting down there. So primary production on the rocks and things like that wasn't really happening much. The bacterial biomass, clear difference between the reference and the treatment over time. So a lot was happening to the microbial community. Over here is photosynthetic production on the rocks. It went up, but then when the Calamagrostis got tall, it went down. 10 minutes. Ooh, gotta go, Andy. Thank you. Here's a really incredible result. So this shows the invertebrates in the reference reach and the treatment reach in July and August. And I'll just take you down here to the bottom right away. In the treatment reach, the abundance of invertebrates on the rocks and under them increased orders of magnitude, way up to over 55,000 organisms per meter square. The fish responded. What happened was the little fish basically moved up to the head of the place we were doing the dosing, and there were more of them, and they were bigger, and that's what's shown in this diagram. And they grew faster, and that's what's shown in this diagram. So the fish were like almost instantly responding to this increase in invertebrate resources. They just hurried up there and they grew faster and bigger. We did some wetland studies too. I'll go through this quickly. Sorry, Andy. Thank you, Andy.
Yeah.
So this is basically, does it matter whether the alder's up here on the hill or whether the alder's down here? And the hypothesis was that if you had alder near the stream and far or no alder, if you fertilize the wetland here at the creek margin, where you had no alder, you would get higher productivity response than you would if you had alder. That was the hypothesis, and that's basically what was shown there. So that the alder is an important source of nitrogen to the wetland. And then we also did a sort of a larger wetland study where we fertilized and did not fertilize and measured a whole bunch of processes, decomposition using tea bags. This shows a range of the things we did. We put pots of Calamagrostis in the creek that had alder, creek that had no alder. We collected soil samples and extracted the soil water to measure nitrogen. Here are some results from the cores. Clearly where you had alder at different depths in the soil profile, you had more nitrogen compared to the no alder. If you looked at ammonia in the interstitial water, same deal. You had much more where you had alder compared to not having alder. And this sort of summarizes what happened to the wetland there. So if you have a site that has alder, you have more nitrogen per unit area than you do where you don't have alder. You have more Calamagrostis at the creek bank, hanging over the creek bank. But here was a really interesting result. Where you have nitrogen, the plants don't need to go find the nitrogen anywhere, so they have much less nitrogen in, they have more nitrogen, they have more roots where you don't have alder because they got to explore the environment to find where the nitrogen is. Had not expected that result. And if you look at biomass, you basically see the same pattern. A real difference between where you have alder and where you don't have alder. So we're getting there. So here's kind of what we think it looks like now. You have alder on the landscape, surface water flowing down, subsurface water emerging at the creek bank to support the productivity of the wetland, which has its interaction with the fish. And then in between there, you may have these carbon-based systems that are adding carbon to the system. So this would be an ideal hotspot on the landscape where you had those two things. So what's next? Does any of this mean anything other than a bunch of publications? The answer is yeah. So what we're interested in is Ryan King going back and catching more kings. That's the deal. And so we had a whole series of things. We met with the Ninilchik tribe, the local tribe that owns much of the land that we worked on. And they had this potlatch where we went and ate and heard talks and everything. And over here on the right, this guy, Artis, he made this alder tree on the right, which is made out of metal. Here are the leaves, and down here you can almost see them. They're little dots or little bacteria in there that fix the nitrogen. So we met with them, and Argent and his people are now using that as part of their education of their young people. We took groups out to these sites and let them see baby fish. Here is Malcolm Milne. He's president of the North Pacific Fisheries Association. They saw this. They had never seen these baby fish. They ended up taking their money and giving it to the land trust to buy land in an area which our models had identified as being important for fish habitat. So that was a positive benefit. I pretty much backed out of the project for reasons, but Mark Raines, shown here, and his wife Kai, they're up there doing groundwater stuff because the groundwater is really important, either as springs coming out. There's one really flowing over here. And this is kind of their view of what happens now. You have water penetrating, flowing over the surface, penetrating, meeting different layers of resistance, and then flowing out and having these springs which appear along the hillside or come out near the stream itself. Well, is this important? The answer is absolutely. What you're looking at here is a graph of air temperature over time. And this is 0 degrees. And the black line up here is groundwater. So even in a cold, cold winter, when everything's frozen up there, this groundwater is emerging in these tiny creeks, creating little areas where it's not frozen, and that's where the fish are surviving the winter months. So groundwater is absolutely essential for these systems. He's taken this further. He does modeling, and he's taken the whole Kenai lowland area, and he's produced a model based on the geology and the topography, which looks at recharge areas, where water's coming back. And he's applying that, and we're doing things with middle schools up there, and they've actually changed the laws now so that they're protecting the streams, and here's just some examples. Protecting the watershed that is water for Homer, and we're almost there, Eddie. So a whole bunch of people have been involved in this, and I was going to talk about orchids, but I'm not going to talk about orchids. Don't have time. But I do want to say that this is incredibly sad because the daughter of this music professor from Maryland, she contracted cancer and was dead within 3 months. It was heartbreaking. She was an incredible woman. Miss her terribly. So I've had a lot of fun up there for about 20 years. I'm not a big salmon fisherman, but I can catch the little ones. And I love going there, and I hope that I convey to you that some of the research we can do here in the Bay can have meaning elsewhere in our world. So thanks for hanging out.
I'm sorry, Dennis, that our time thing here, but
Okay.
let's see, we got 5 minutes for questions here though, so that's great, awesome, that's amazing, Dennis.
It's fun. I will do orchids in. I came up with the idea of paper punch-out models of native orchids. We have 30 of them. I can give them to you in small numbers, but Christmas is coming, and we have a box set of 20 models with a booklet in here about native orchids, and if you contact me and send me $20, I'll send you one of these great Christmas presents.
Are they still on Amazon?
Still buy them from me because $20, Smithsonian gets $10. If you buy them from Amazon, you don't get much at all. So you can buy them on Amazon. Amazon. Yeah. Okay. So questions about Alaska? Yeah.
I think it's really nice to be pointing out the subsidy stress relationship between the nitrogen and the productivity. And I was wondering what your thoughts were if you were actually going to add even more nitrogen, and at what point do you continue to get more productivity at some point, or at some point, that becomes stress?
Probably what happens in areas where you have a lot of alder on the landscape, they're getting lots of nitrogen. Those systems are carbon limited, so another nutrient sort of kicks in that controls, fish production, how many fish are there. And that goes back to the notion of Ryan's modeling, looking at alder landscape features and being able to predict where hotspots were, where you had just the right amount of carbon coming in, and the right amount of nitrogen, and that's where you would find most baby fish. And we went out and tested that, and that worked. Yeah.
Dennis, really enjoyed your presentation. I'm wondering, are there thoughts or perspectives on sort of what affects where the alder, the abundance of alder are in the landscapes? Any interest, intention about sort of trying to alter that or plan it? Or is this sort
Yeah.
of more descriptive in that?
Yeah, one of the spin-offs of this is when they do a little bit of stream restoration up there where they have erosional issues. And before this work, they were planting willow because moose like to eat willow and they don't like to eat alder. But they changed it. They're planting alder in places where they used to plant willow. So that's sort of a practical side of it. But the alder story is a really important one up there because with climate change, there actually have been insect outbreaks that have killed alder. And that led to that sort of one study we did, in fact, one I'm finishing up now. But as the climate warms, the alder are moving further north in Alaska, so they're going to alter the systems. I don't know if anybody's actually looking at that yet, but that's a big.
You said at the beginning that they protect streams that are known to have adult salmon, but after your findings, did they decide to protect the upstream regions that hold the salmon oils?
Yeah, and that got at the last slide, basically, because I didn't have time to really go over, which is because of this work, they're now having people they can't get too close to a stream if they're doing a sand and gravel operation. We've had landowners that we've met through various conversations say, I want to do X, Y, and Z on my land. And we can look at that now and say, okay, where is that relative to the stream? What's the landscape configuration? We actually had people change what they were going to do to a different location because of the effect that they might have by removing the alder, for example. And the city of Homer, I mean, half of their water is coming from those headwater streams and the groundwater. So they are being, and so what's the link that's going on there is the Kachemak Heritage Land Trust, which does the same thing that land trusts do here, they save land. They're focusing much of their efforts now on places where, one, they're protecting streams, but they're protecting resources that, benefit the stream too. But that's, when you think about it, that's a big deal in Alaska. Alaska's not Maryland. I mean, they don't like regulations up there, they don't like laws up there, they don't like, they want to do what they want to do. This is beginning to have, some effects. I think it's pretty cool, isn't it?
All right, well, thank you so much.